For lecture 3 in the course, we're going to talk about measuring digital gene expression. Remember, RNA-seq data is count-based data. We're counting the number of reads that map to a particular transcript, and so it has a digital quality to it. And that means we need to consider the kinds of units that we can use for quantifying the expression of a gene. And that really is at the heart of a larger process that we'll spend quite a bit of time talking about today, which is normalization. Normalization is essential for analysis of RNA-seq data, but it's also a general process that you can go through with many different kinds of genomics data sets. All right, so before we dig into normalization, let's just click on the lecture card for today and quickly take an overview of our learning objectives. We'll begin by just reviewing the steps from the last class. So using Callisto to create an index from a reference transcriptome file, and then mapping raw reads against that index to quantify gene expression. We'll then actually look at the outputs that Callisto produces, and that gives us a chance to actually view those units of measurement that I just referred to, and it also really starts to set a stage for this what will be the first half of the lecture in talking about this normalization process. In the second part of the lecture, we'll begin the RStudio project that we'll use for essentially the remainder of the course. And as we do that, we'll open our first R script. We'll take that opportunity to kind of take a step back and discuss the anatomy of an R script, if you will, and also start to install the R packages that we'll use to work our way through that first script. Now, this marks a really important transition in the course. Up to this point, we've been working with command line software tools that we accessed using the terminal on a Mac OS or the command prompt on a Windows machine. And now we're going to leave that Bash environment and we're going to move into RStudio. And we'll stay in RStudio almost for the rest of the course until close to the end when we need to return to the Bash environment for actually running some command line software for handling single cell seq. Now, when we open this first R script today, you'll notice that the entire analysis of RNA-seq data throughout this course is broken up into steps. And these steps make it easier for us to talk about discrete stages in the analysis in a really concise and controlled way. And so anytime there is a piece of code associated with a lecture, it's actually available for download on that lecture page. But I also want to take just a minute to make sure everybody understands where to get these R scripts. So here we are on the lecture page. There is that step 1 script. You should be able to just click on that and download it. But I wanted to point out that at the top of the page, there is a scripts tab that when you click on it, will take you to a page where all the R scripts are kept. And you can see step 1, step 2, etc. Now, there are a few important things to point out. The first is this disclaimer at the top of the page. Sometimes, depending on your particular browser or the way your browser is configured, clicking on one of these links actually won't download the R script, and you may think that the link is broken or that we don't have the scripts actually posted to the website. That's not true. In fact, my web browser does that exact thing. If I click on this, I get a tab that briefly opened and then closed down. So if that happens to you, I've described what you should do here. You may try a different browser. This is Firefox. Clicking on that exact same R script now opens a download window. Or alternatively, you can Control or Alt click the link and scroll down to copy link address. And when you do that, you can paste that URL back into the browser, and that should successfully open that download window. All right. When you download this, it'll be an R script and it'll be something you open in RStudio. We're going to go over that later. I just want to make sure everybody understands how to access these R scripts. Okay, heading back to the presentation. So that's really where we are today. So let's do a quick high-level overview of what we've accomplished so far and how it relates to an RNA-seq workflow. So this is a schematic that we'll kind of build on as we go through the course that takes us from raw data that comes off of a sequencer and our reference files that we get from a reference genome resource, and we move through a process of starting to do read mapping. So what we've done thus far, and what we covered in the last lecture, is using command line tools like FastQC or FastP, using multi-QC to assess the quality of the raw reads that we got from the sequencer for doing a pretty comprehensive QC or QA analysis. We then used Callisto, and particularly the index function, to take an organism transcriptome reference file, a FASTA file, and turn it into an indexed transcriptome. And we did that using this simple bit of code where we provided the name of the input FASTA file and the name that we would like of the index we'd like that to be structured into. And we spent some time in the last lecture talking about some of the key concepts involved in constructing an index, like a Burrows-Wheeler transform, and how these methods allow you to create a file that is fundamentally easier and faster to search against. We then used Callisto, but this time the quant function, to take our raw reads that we had and map them against this transcriptome to get summarized read counts. We did that using some code that we spent some time talking about, and we also ran on a subsample of our raw data so that it could run more quickly. And then in the lab, if you were able to attend, we spent some time really going over how to automate this process so that we could do this for many samples using either a shell script or a for loop. All right, in today's lecture, we'll open the step 1 script that'll pick up where Callisto left off. It'll read in the Callisto data, and it'll move us through here. Now, we won't cover that full process today. Today, we'll focus on getting you set up in RStudio and making sure you understand what these scripts are and how they work. And then in the lecture next week, we'll actually get down to the business of running the code in the step 1 script. So I want to take this minute to highlight something really, really critical. And that is, if you were unable to do this QA analysis, and or if you were unable to index your FASTA file or map reads against it with Callisto, don't worry. You are still able to continue for the rest of the course. Remember, we've provided multiple on-ramps for you to be able to continue along this process of analysis, and that's all pretty clearly laid out on the Data tab. If you scroll down, we hope that many of you could process raw data on your laptop, but we provide this second option, which is we've made those Callisto output folders immediately available for download. They're small in size, so there should be no problems with just essentially pretending as if you did carry out the read mapping on your laptop. Okay. Now, I just want to take this minute because we'll be leaving Callisto behind after this, and we really won't return to it until we use Callisto in a bit of a different form later in the course. So this is just a good point to highlight that there's a lot about Callisto that we didn't have a chance to explore yet, but that you may want to take some time to look at on your own. For example, the quant function has a number of different options that we could have provided to it. One of them, pseudo-BAM, will allow you to essentially generate BAM files from these pseudo alignments that can then be used for representing your data in these sort of pile-up plots on a genome browser. And then you've probably seen these on genome browser tracks or in presentations where we have a reference gene in this case, and we've piled up reads along the various exons of those genes so we can understand expression data in a pretty visual way. All right. You also just might want to be aware that if you're interested in other aspects of the transcriptome, like long non-coding RNAs or link RNAs, there's some recent data that suggests that Callisto and pseudo-alignment tools like Callisto are particularly good at detecting long non-coding RNAs, and that's important because link RNAs are often some of the most weakly expressed genes and some of the most weakly differentially expressed genes, so they sort of serve as a good benchmark to understand how good a read mapper is at really detecting and quantifying these kinds of difficult to measure genes. And so as they conclude in this study, we compared the performance of pseudo-alignment methods like Callisto and Salmon and alignment-based methods like HT-seq, feature counts, RSIM in link RNA quantification by applying them to a simulated RNA-seq data set. In summary, pseudo-alignment methods like Callisto or Salmon, in combination with the full transcriptome annotation, is our recommended strategy for RNA-seq analysis of long non-coding RNAs. Okay, so If you want to keep working with Callisto, as I said, there's a lot that we didn't cover. We didn't do paired end read alignment. We didn't generate these pseudo-BAMs. We've laid out a protocol on the protocols website that you've already gotten used to using. So if you go to this protocolshostmicrobe.org compute Callisto, you'll see that protocol here, which tells you or reminds you how to build an index, how to quantify reads against that index, how to handle paired end reads, and then how to think about stranded alignments and creating these kind of bigwig files that can be viewed in a genome browser. Again, this is beyond the scope of certainly today's lecture, but really the course as it's structured now. So this is stuff you could explore on your own, but just be aware this stuff may require more resources than you have available on your laptop. Okay, so and one last thing to just touch on is that Callisto does have a unique function for handling single cell data. And so I mention this because we will come back to Callisto for a more recent development with this software, which is the ability to process large amounts of single cell data very, very quickly. Okay, so why don't we now go ahead and open up our project folder. I'm just going to close out Firefox here. Okay. Now, depending on how you've structured your project folder, I've got my raw FASTQ files in one folder. I've got those downsampled FASTQ files in another, and I've gathered up all my Callisto outputs into another folder. And you can see each folder here corresponds to the read mapping results from mapping a single sample against that reference transcriptome. And as we discussed in the previous lecture, I've also generated a log file that contains information that would have been printed out to my terminal, but that I wanted to capture so that MultiQC could parse it. So I just want to open one of these folders. I'm going to choose the top one here, which is one of the patient samples, patient number 8. And when I'm in this folder, what I can see here is I really have 3 files. I have one that is an abundance.h5 file. I have one that's an abundance but a TSV, a tab separated values file. And then I have a JSON file. All right, the JSON file, just so you know, if I drag and drop that onto Sublime, I can see that that's really a log file in and of itself. It contains information about the number of reads processed, almost 95 million. So this particular sample was very deeply sequenced. And I can see how many reads pseudo-aligned. So about 62 million, 61 million reads pseudo-aligned. And conveniently, it's also stored exactly the command I ran to do that alignment. So that's always kind of preserved there, which is great. And of course, I know the date that this read mapping was carried out. So you may be wondering if I get all this sort of log information for free, why on earth did we generate our own log file? And the answer simply is that MultiQC can't parse these JSON files. So we want to generate a .log file equivalent to this that MultiQC can handle. Okay, so you now have that log information stored here as well. You may or may not, depending on how you ran Callisto, have this abundance.h5 file. If you don't have it, don't worry. It's not going to be so critical for moving through the course. What's critical is that you have a TSV file with your abundance data. Okay, so I'm going to drag and drop this on Sublime. And when I do that, I can see the structure of a read mapping result of transcript quantification for this one sample. All right, so I have what appear to be 5 columns here, right? I have a target ID, and that is really the transcript identifier. These are ensemble transcript IDs. They have these sort of miserably long numbers, and ENS for ensemble, transcript T, and then 5 or 6 zeros here, and then a number. These transcripts can have different versions, and those versions refer to different annotation versions, updated reference sequences or information about the transcripts. So they can also end in a .1 or a .2. But the real information here for the transcript ID is this bit I've highlighted. We also have a length. So in this case, the transcript was only 12 nucleotides long, but if we scroll down, we can now see transcripts that are more what we would consider normal in terms of their length. We have an effective length, as well. And if you're wondering about what length and effective length are and how they're different, I'm going to talk about that in just a minute. We then have the estimated counts, and the estimated counts are the number of reads that mapped to that transcript. So we can see that at the top of our view here are a whole bunch of really small transcripts for which no reads map to them. And then finally we have this TPM, and this is the unit of measurement that we're going to spend some time unpacking. It's transcripts per million, and it's a critical way that we can think of representing and quantifying gene expression data. I'm going to scroll down just so you can see some of these TPMs here. So here's a transcript. I have no idea what this transcript is, and we'll get to that in a later lecture about how we annotate these with other information, like which gene symbol, what gene that transcript comes from, what kind of gene it is. But right now, all we know about it is this transcript ID. We know its length is 652. Its effective length is 403. Those effective lengths will always be smaller than the actual lengths. Again, we'll get to that. We know the estimated counts, about 115 reads mapping to that transcript. And we get that data expressed as a transcript per million, or TPM. And this transcripts per million simply is actually quite easy to understand. What will take some time to go through is how it arrives at this number. But it is telling us that if we sampled 1 million transcripts from this patient sample, 5 of those 1 million transcripts would be coming from this gene. Okay, so what is that gene? You could always search that transcript identifier, and we will hit on that gene in Ensemble. Okay, we're not going to take any time to really search and cherry pick any particular genes here, but I just wanted to kind of highlight that these are genes that we could identify. Okay, so what we're going to do in the next part of the lecture is actually talk about this effective length versus length, and then we'll get into how we get from these read mapping estimated count measurements to transcripts per million, and why it's important to think of our data in that kind of unit of expression. Great.